Diese Konferenz wird nun aufgezeichnet.
So, welcome everybody to our BIH lunch lecture of today. And it is my great pleasure really to introduce today's speaker, which is Professor Aroon Hingorani from University College London. He's a professor there of genetic epidemiology and was acting as formerly as a director of the UCL Institute of Cardiovascular Science, and also a program lead there for cardiovascular research there at the UCL hospitals. And although his work focused primarily around cardiovascular diseases and sciences, the research group now centers around discovering genotype-phenotype relationships, really with the aim to identify and validate novel drug targets which help us in the treatment of many diseases. And with this, I think his research focus and also achievements very well fit to the overall aim of the Berlin Institute of Health and also the Charité. And that's why it's really great to have him here today to give a talk, a presentation. And I hope we really engage on a lovely discussion afterwards, and also have further contact with him in the future. So thank you very much, Aroon, for accepting the invitation and being our speaker today.
Thank you very much for the very kind introduction and for inviting me to present this morning. Can I just confirm that you can hear me clearly and that you can see my slides?
Absolutely, yes.
Thank you. Okay, so my lecture is entitled Democratising Drug Development Through Human Genomics. I hope that the reason for the title will be clarified by the end of the lecture. But just to orientate you, first of all, I'm going to divide the presentation into three parts. In the first section, I'll say something about the problems that we face in drug development. In the second part, I'll say something about how human genomics can help address some of those problems. And then in the final part, I'll say something about the implications of a genomic-based approach to drug development, really for the next translational phase of human genomics, but also for the whole way in which we develop drugs as a society and the socio-economic aspects of that. So to start with problems in drug development, I think this audience will be familiar with the idea that there is a very high failure rate in drug development. Many reviews of the field have suggested that perhaps 96% of all drug development programs fail. And this has a number of consequences. Firstly, for healthcare systems and patients, it means that the cost of any successful drug is set to recoup the cost of any prior failure. And this means that new drug costs are often very, very high. For example, in 2018, the top selling drug by dollar revenue was adalimumab or Humira, and the revenue generated by that drug was over $20 billion in that year. And so the price of new drugs is really very, very high indeed. And that cost has to be borne by healthcare systems. And in a public healthcare system, such as the one that we have in the UK, those costs have to be met by either raising taxation, by diverting funds from other sectors of the economy, or by limiting access to the less cost-effective drugs. And we have an organization that oversees that called the National Institute of Health and Care Excellence, or NICE. But the high failure rate also has consequences for the pharmaceutical industry itself. It tends to engender risk-averse research and development, such that we often have many drugs against the same target and mechanism for the same disease, many so-called Me Too drugs. There are motivations because of the high failure rate to extend the patent life of drugs, so-called evergreening. And what this means is that we tend to have a very patchy therapeutic armamentarium for many diseases. So some diseases are very well served by therapies, but others we have no good treatments at all. And also for the pharmaceutical industry, if an area of drug development fails frequently, it can result in job losses and actually a diversion of research and development into other sectors. And we've seen this at various times in neuroscience, even in cardiovascular disease. So the question arises, why is the failure rate so high? And again, many reviews of drug development over the years have come to the conclusion that one of the major reasons for failure is a surprising one, actually. It's lack of efficacy of the drug in the intended indication. And this is frequently unveiled at the final step of drug development, at the time of the phase 3 randomized controlled trial. And it's not restricted to any particular disease area. This is a feature for drug development in any of the major disease areas. So drugs fail all too frequently for lack of efficacy in the intended indication. So this means that something must have gone wrong in the early phase of drug development. So perhaps it's worth spending some time just thinking about how that might happen. So essentially, almost all drug targets are proteins, and in drug development, there are several key tasks. The first is to try and identify which proteins are important in the pathogenesis of which diseases, and if any of those proteins are druggable. By that I mean, are they readily accessible to small molecules or monoclonal antibodies or peptide therapeutics that might modify their action to mitigate their effect on disease? So once a protein target has been identified and a compound has been identified, then the steps in drug development involve proving target engagement and then proving safety and efficacy in clinical trials. But if you get the first step wrong, if you work on the wrong protein target in the wrong disease, you're effectively destined to fail. So could that happen? And does it happen frequently, and why? So I think there are two flaws in drug development that relate to matching the correct protein target to the correct disease. The first is that traditionally, drug development has focused on identifying and validating therapeutic targets, proteins involved in disease pathogenesis, in cells and tissues and model organisms. And they have turned out to be an unreliable predictor of treatment efficacy in humans. Moreover, those sorts of systems produce a capacity only to test a few drug targets at a time. And the third element relates to this reproducibility crisis that we hear much about in preclinical, but also in epidemiological research. And that is this high rate of false discovery, that many research findings may be false. And collectively, if these sorts of pieces of evidence from preclinical discovery are taken into clinical phase testing, there's a high likelihood they might be wrong. And you only unearth that when you do the definitive test in drug development, which is the randomized controlled trial, the final step. So to summarize, preclinical studies have issues in relation to external validity and reproducibility, but even human observational studies that might inform which targets to work on are prone to difficulties of confounding, of reverse causation, and also reproducibility. And this is compounded by the fact that there may be very few protein disease causal relationships to discover. So one way in which we've been beginning to think about this is to consider a matrix shown on the left-hand side of the slide, in which we consider a sample space where diseases are the columns, if you like, and genes or proteins are the rows. And one can start then to parameterize this space with a thought experiment and ask the question, well, how many diseases are there? Well, let's imagine there are 10,000 complex human diseases. We know the number of protein coding genes in the human genome from the Human Genome Project, and it's around 20,000. So that means there are about 200 million unique protein disease pairings in this sample space. And then we can ask the question, well, how many causal genes or proteins might there be for any disease? And if we consider there may be 100, then we are effectively looking for 100 times 10,000 or 1 million causal gene or protein disease pairs within this sample space. So if we were to put our hands into the sample space and pull out a protein disease pairing at random, our probability of selecting a causal gene or protein disease pairing within the sample space is about 5 per 1,000 or 1 in 200. So the prior probability of working on the right protein disease pairing is small. Now, of course, you might say, well, we don't just pick targets and diseases to work on at random. We actually rely on previous medical research to prioritize which areas we might choose for drug development. But as John Ioannidis and others have noted, there may be a high false discovery rate in biomedical science. So, for example, if there are 1 million out of these 200 million causal pairings to detect, but our experiments have a power of 80%, we would only identify 800,000 of those million. But more importantly, if we operate at a false positive rate, an alpha of 0.05 in our experiments, as is typical in biomedical research, because there are so many non-causal pairings, 5% of the 199 million non-causal pairings is very much larger than the 80% of the 1 million causal pairings. And therefore, the experiments we declare as positive may be more likely to be false discoveries than true discoveries. And in fact, we can consider preclinical evaluation, if you like, as a diagnostic test for future drug development success. If we take only the positive successes from our preclinical experiments forward into clinical phase testing, but most of those positives are false positives, then we have a very high false discovery rate in clinical development and a high failure rate. And we can parameterize this as an equation of drug development success, if you like, which tells us that our likelihood of success is based on the number of true relationships available for discovery, which may be small, and then the power and the experimental false positive rates in preclinical and clinical phase testing, which may be very high. So how can genomics help reduce this high failure rate in drug development? Well, the principle which can be applied is that given that most drug targets are proteins and proteins are encoded by genes, we can use the principle that natural genetic variation in genes that encode a drug target, whether the variants are common or rare, whether they are coding or regulatory, if they affect the expression or the activity of the encoded protein, and that protein plays a role in physiology, metabolism, and disease, then evaluating the genetic associations with physiology, metabolism, and disease of variants in the gene encoding those proteins could help us anticipate the effects of a drug that targets the same protein. And the advantages there potentially are that the experiments and the analyses and the findings that we have come from the correct organism, that is the human being. If we use genome-wide association analysis to look at variants across genes encoding all potential drug targets, we can test them concurrently in parallel. We don't have to select a subset of targets. We know that in genome-wide association studies, they are designed with a sample size and with replication such that there is a low rate of false discovery. But a further attribute is that variants in a gene encoding a drug target and variants in a gene in general are inherited by a randomized allocation at conception, according to Mendel's laws. So genetic studies are, if you like, a type of natural randomized controlled trial, which emulates the design which we've come to be used to as the pivotal experiment in drug development, which is the randomized controlled trial. So collectively, genetic studies have some potentially beneficial attributes for drug development. So let me return to this sample space of 10,000 diseases and 20,000 protein coding genes and just highlight the top left-hand corner, if you like, of that sample space, the first 100 diseases and the first 200 genes. And let's imagine there are 1,000 causal genes per disease. When we do a genome-wide association study, effectively we're interrogating the causal relevance of all the encoded genes for a given disease, and we might generate some hits. And that might help with drug target identification. So we're particularly interested in proteins that are causal for the disease, but more so in proteins that are not only causal, but also readily targetable by small molecule drugs or monoclonal antibody therapeutics or peptides. And we think of all the 20,000 protein coding genes in the genome, about 4,000 of those encode druggable targets, and they're highlighted as the orange squares in this panel. And so is there evidence that human genomics and human genome-wide association studies could be useful as a tool for identifying drug targets in this way? So I think there are at least 4 lines of evidence that are emerging. When we look at genome-wide association studies, we see frequently that they rediscover known drug target disease indication pairings. There's been work done in the pharmaceutical industry itself to show that there are higher rates of approval for drug target disease indication pairs that had genetic support than those that didn't. There are also many examples arising from genome-wide association studies in specific areas. And now there are even examples of drugs, successful drug development motivated by human genomics. So just to look through some of these points of evidence. So we showed back in 2017 that there's a very high rate of rediscovery of known drug targets by genome-wide association studies, suggesting that if genome-wide association studies uncover new targets, they're quite likely to be successful if pursued for drug development. And examples, come from many disease areas. So, for example, a genome-wide association study in Crohn's disease identified the tumor necrosis factor alpha gene. And of course, that's already the target of monoclonal antibody therapeutics for the same disease. And there are numerous other examples from other disease areas. So this is just showing some of the evidence from the pharmaceutical industry that they are noting higher rates of approval for drug target disease indication pairs that had prior genetic support. And this is 2 independent pieces of work from different companies. And we see findings again and again from genome-wide association studies showing the uncovering of drug targets by this technique. Here's an example from rheumatoid arthritis, identifying known drug targets for rheumatoid arthritis, either directly or by the protein binding partners of some of the proteins identified as being causative from these analyses. Example being here, for example, tocilizumab, which targets the interleukin-6 receptor, tocilizumab already licensed for rheumatoid arthritis. Now, of course, genome-wide association studies highlight regions of the genome that contain causative genes, but sometimes it can be challenging to identify which particular gene is the causal one, particularly if there's extensive linkage disequilibrium across a locus and that there are many genes lying within that region. But there are a number of emerging methods that are either statistical, that leverage expression data, functional annotation, or orthogonal evidence that are helping in this gene prioritization process. But one interesting insight that's emerged recently is when you compare features of genes, when you have a truth set in a region, so that you already know the causal gene in a region of a signal from a genome-wide association study, what are the features of that gene that indicate that it's likely to be causal? Well, actually, the most important feature it turns out to be is its proximity to the strongest GWAS SNP. So actually proximity to the SNP turns out to be one of the most important features. So with this in mind, it's now possible to harness the collective data from genome-wide association studies, as well as genetic data from biobanks, to map these genetic association signals to the causal gene, to the protein encoded by the causal gene, and then through other approaches and other databases to compounds, including licensed drugs, that target those proteins. So effectively to match compounds targets to diseases, which is essentially what we want to do in drug development. So this is a process that we've undertaken, but there are also other very important collaborative efforts, such as Open Targets, a partnership with EBI, the pharmaceutical industry, and the Wellcome Trust Sanger Center. So I'm going to say something now about target validation as opposed to simply target identification and how genomics can help with that. So coming back to this principle that there's a convergence of the effect of genetic variation and drug action on proteins, one might hypothesize that it should be possible not only to map which proteins are responsible for which diseases and therefore which are the most useful drug targets, but also to gain insight into what the perturbation of a target might do to physiology, to metabolism, to imaging traits, to function, as well as to disease risk. So what's emerging, for example, is that when you piece together information from many genome-wide association studies, you see that the same target appears as an important locus for more than one disease. And that is, one target can affect many diseases and many different outcomes. And so a new type of study design is coming to the fore, and that's the phenome-wide association study, where you test the associations of a single target against many biomarkers and many disease endpoints. And that gives you really a readout of what a drug might do if it acted on the same target. So here's an example in relation to disease endpoints. So I mentioned genetic variation in the interleukin-6 receptor before. So in a GWAS of rheumatoid arthritis, variants in the gene encoding the interleukin-6 receptor were identified as being causal, and we know the interleukin-6 receptor is a target for a drug, tocilizumab, that is effective in the treatment of rheumatoid arthritis. But the same variants in the interleukin-6 receptor seem to confer protection from coronary heart disease, from atrial fibrillation, from abdominal aortic aneurysm, and from type 1 diabetes, but might increase the risk of asthma. So you can see how you can begin to picture a readout of the effects of a drug acting on a given target on multiple diseases at the same time. And in fact, there are now clinical trials underway to explore the potential to repurpose tocilizumab as a treatment in coronary heart disease. And not only do you see this effect on disease endpoints, but also on biomarkers. So this study compares the effect of variants in the gene encoding the target for statin drugs, which is HMG-CoA reductase, with the effect of taking a statin on several hundred metabolites measured in a blood sample. And if you look in the right panel, you can see that the variant in the gene produces a rank order of effects on a whole range of metabolites that is exactly reproduced when you take a statin drug. So the same metabolites change in the same rank order. So you could do a thought experiment that if you hadn't already invented statins and used them, that the genetic findings for the effect on the metabolism give you a very faithful readout of the effect of statin treatment on the metabolome. So we've begun, and many others also have begun, to use genomics as an important tool for target validation and to use the approach to start to address important drug development questions. For example, to distinguish on versus off-target effects of drugs, to identify repurposing hypotheses, and to provide genetic support for novel drug targets and to help understand the likelihood of success or failure in a clinical trial. So the principle that I've described actually is really a special case of something you may have heard of in a more general epidemiological context, which is the principle of Mendelian randomization. And Mendelian randomization is a principle that's been applied primarily to address problems that arise in observational epidemiology of confounding and reverse causation. So if you have an exposure of interest, and an exposure could be anything from a protein concentration to a biomarker to blood pressure to even a behavior or an attainment like educational attainment. If you're interested in the association between an exposure and an outcome like a disease, if you just do standard observational epidemiology, any association you discover could be prone to confounding by an unmeasured or unknown factor or reverse causation. And the use of a genetic instrument that proxies the exposure can provide a route to overcoming confounding and reverse causation because of these attributes of genotype, randomly assigned, which eliminates confounding, and because there's a unidirectional flow of information from genotype to exposure to disease, this overcomes reverse causation. Now, the Mendelian randomization paradigm has two assumptions. The genetic variants that one uses to instrument the exposure of interest should not have any direct pathway to disease independent of the exposure, either through the confounder or directly in some other way. So these sorts of bypass pathways are referred to as pleiotropy. So we need to be clear in Mendelian randomization work that we're trying to minimize and avoid pleiotropy because we may make incorrect causal inferences on the relationship between an exposure and an outcome if we don't. Now, this sort of thinking, I think, is very relevant to the use of Mendelian randomization and genomics in general for drug target validation. And it sometimes leads to a little bit of confusion, which I think it's maybe important to highlight, but also to address. So in cardiovascular disease, important questions emerge, such as, is a particular biomarker like high-density lipoprotein cholesterol causally linked to heart disease? So that's a risk factor-focused question. It may lead to a drug development hypothesis around HDL cholesterol, but it is subtly different and importantly different from a question such as, is cholesterol ester transfer protein, a particular protein that might be a drug target for heart disease, which affects HDL cholesterol, asking the question, is CETP causally linked to coronary heart disease, which is a drug target focused question. These two types of questions can both be addressed by Mendelian randomization analysis. However, the technique that's used is different. In the case of high-density lipoprotein cholesterol, the genetic instruments that are used are typically selected from throughout the genome, leading to genome-wide Mendelian randomization. Whereas if your question is about a particular drug target, the optimal genetic variants to use are those that are within the gene that encodes the drug target itself. So these instruments lead to a type of Mendelian randomization analysis, which we are calling cis Mendelian randomization analysis. And you can compare the outcomes of these different types of analysis. So the top panel here shows the result of a genome-wide Mendelian randomization analysis of HDL cholesterol on coronary heart disease, the suggestion of a protective effect, but some uncertainty around it. The middle panel shows the results of a drug target cis Mendelian randomization analysis of variants in the CETP gene, and one can see strong evidence of a causal protective effect on coronary heart disease. And in fact, we now know that that effect is a real one because specific potent drugs like anacetrapib that target cholesterol ester transfer protein in clinical trials are protective from coronary heart disease. And one can see that the effects through a druggable target may be diverse, and it's quite difficult to judge what the mediating factor might be. So what I show on the top panel here is the effect of variants in the CETP gene, either on LDL cholesterol on the left, HDL cholesterol in the middle panel, or triglycerides on the right, and concurrently the effect of those variants on coronary heart disease risk on the y-axis. So in the top panel, you can see that variants in the CETP gene affect LDL cholesterol, HDL cholesterol, and triglycerides, and all of those are associated with an effect on coronary artery disease risk. Whereas variants in the lipoprotein lipase gene, which also influence coronary artery disease risk, appear to do so independently of LDL cholesterol, but potentially through HDL cholesterol or triglycerides. So there can be complex mediating effects downstream of the protein. So what this centers us on now is this idea that the optimal exposure in a drug target Mendelian randomization analysis is a protein encoded by the gene in which one uses variants as genetic instruments for these analyses. And this has a number of advantages, one of which is that almost all drug targets are proteins, and therefore proteins are an exposure of interest in drug development. But also because proteins are a very proximal consequence of genetic variation, just one step down from transcription, it means there is less potential for pleiotropic pathways between genetic variation and disease to operate independently of the encoded protein. And what this means is that Mendelian randomization analyses of protein exposures as drug targets tend to adhere to the important assumptions of the Mendelian randomization principle, more so than more downstream exposures of interest, such as biomarkers or even things like educational attainment. So in fact, when we repeated the Mendelian randomization analysis of CETP, this time weighted by the measures of the CETP protein itself in the plasma, we see very robust effects, same as we saw when we were weighting by a downstream effect on HDL cholesterol. And we see in a phenome-wide association analysis that CETP genetic variation confers protection from coronary heart disease, but also from heart failure, from chronic kidney disease, but actually potentially increases the risk of age-related macular degeneration. So this type of readout informs what a drug that targets CETP might or might not do. Now, there are some emerging important technological advances that means that Mendelian randomization analysis with protein exposure is becoming much more feasible. And that's the invention and development of technological platforms to measure very large numbers of proteins in the serum and plasma. And two frontline technologies come from SomaLogic and Olink. And already analyses are being undertaken to identify genetic variants that influence the level of these proteins. Here's an early example from Sun et al. Published in Nature, but more recently, very large-scale analyses of the consistency and agreement between these platforms conducted by Mike Pietzner and Claudia Langenberg have been published from Berlin, from Charité, and also very large-scale genome-wide association analyses of the circulating proteome, identifying both cis and trans variants that identify the levels of these proteins, which now provide really powerful genetic instruments for drug target validation. And this paper was published by Mike and Claudia in Science last year. So as I mentioned already, the optimal genetic instruments when the protein is the exposure of interest, as it is in drug target validation, are variants that lie in and around the encoding gene acting in cis, hence the term cis-MR. Although we frequently find variants outside of the encoding gene that can affect the level of the protein of interest, these trans variants can sometimes be prone to horizontal pleiotropy and undermining some of the assumptions of Mendelian randomization analysis. For example, if we were to use a SNP in a protein outside the encoding gene, where P1 here is the protein of interest, although these trans instruments might be valid instruments for the causal relevance of P1 on a disease, if the pathways, as I've shown in the middle panel, there's also the potential for pleiotropy, where P4 has a direct effect on the disease outcome, P4 instrumented by this trans variant in gene 4 here. And the protein of interest actually has no causal influence on disease. So we prefer, in our analyses of drug target validation, Mendelian randomization of proteins, to use cis-MR wherever possible. So with the advent of proteomics, but also metabolomics, transcriptomics, and imaging data, each on a large scale linked to genome-wide association data, it becomes possible to use this kind of principle effectively to start to generate wiring diagrams of disease, looking at the effect of a genetic perturbation on the proteome, the metabolome, the imaging trait, and the diseasome all together to map a pathway from genetic variation to disease, and through that to draw an inference of the effect of a drug targeting a protein on these downstream variations and parameters. So I think very high potential to take this approach forward at very large scale, looking at multiple targets and multiple diseases in parallel. So I'm going to close the lecture by just considering some of the implications of this approach, whereby genetic information linked to clinical and research-based data in human beings provides the primary source of evidence on drug target development and drug development in general. So just to kind of set the scene a little bit, we think there are around 10,000 complex human diseases. We know that there are around 20,000 protein coding genes in the genome, and therefore a minimum of 20,000 human proteins. More can be generated through alternative splicing and the coming together of multiple protein subunits. We think about 4,000 proteins are readily targeted by small molecule drugs and monoclonal antibody therapeutics. But actually, we only utilize about 680 of those targets by our currently licensed drugs. And perhaps only 1,000 of those targets have been evaluated in clinical phase drug development. So there's a very high potential to work on many more targets against many more diseases. In order to do that, we need to undertake these sorts of analyses at much larger scale. And we need to convert those findings or take forward those findings into drug development. So it was pleasing to us, as we've been working on these sorts of ideas, to see that many drug companies, including here the example GlaxoSmithKline, indicating that they are focusing their drug development primarily on evidence that arises from human genetics, because medicines targeting mechanisms of action with human genetic validation have a higher probability of success. And the way in which this is becoming manifest is by really quite substantial investment from the pharmaceutical industry in data sets that are useful for these types of analyses, particularly very large-scale national biobanks like UK Biobank, but many other examples also exist now around the world, and also in healthcare systems. So, for example, Regeneron has invested substantially in the Geisinger health system in the United States to undertake sequencing and genotyping analysis linked to health record information within the healthcare system. So this is posing some questions about the kind of economic relationships and the flow of money and intellectual property within these sorts of systems. And an example of this comes from the idea that one of the personal genomics companies, 23andMe, which has many millions of consumers that have undertaken a genetic test, personal genetic test for disease risk prediction, but who've also provided information about their healthcare status, recently undertook a contract with GSK, a drug company, for them to mine the data for drug development over a period of 4 years. So one can ask the question that if drugs were developed from that relationship and sold back into the healthcare system, would the citizen or consumer who gave their genetic data and actually paid for the consumer genetic test in the first place risk paying twice, once for the genetic test from the personal genomics provider, and then again for any drug that might be developed from the data that was then passed on to the pharmaceutical industry? I think there's an opportunity here also that healthcare systems might undertake genetic analysis on a much larger scale for drug development and actually enter into much more equal partnerships with the pharmaceutical industry in terms of providing insights that support drug development and reduce the risk of failure, but for doing so, negotiate a revenue stream, pricing and access to those drugs in a much more favorable way than is currently done to mitigate the high cost of drugs in healthcare systems. I think this is something that will need to be considered in the future. So to conclude my presentation, I'll say that the 96% failure rate in drug development, including a huge failure rate in clinical phase testing, is unsustainable financially and has a host of undesirable consequences. There's an emerging, I think, quite compelling case now for using human genomics and linked biomedical data to aid drug target identification and validation. But to really exploit the opportunity of this paradigm requires us to be able to map genotype to phenotype at much greater scale, breadth, and resolution than we're currently doing, although we are on the trajectory to do that. But achieving this level of scale will require, I think, a much larger societal engagement because the data comes from citizens. And it involves healthcare systems, academia, industry, and charities. But if we get the relationships right, this could provide a lever to develop a much more needs-led rather than a profit-led approach to drug development, and hence my title, The Democratization of Drug Development. So I'd like to end by thanking all the funders of this work who I listed in my first slide, and also the very many group members that have basically made this work possible. I've shown them on the left, particularly on the drug development side, Chris Finan, Florian Schmidt, and then a number of the PhD students who I've shown below. And also much thanks to many key collaborators, Claudia, Mike, Ellie Wheeler, Harry Hemingway, and a number of others, and the UCLEB consortium of cohort studies that we work with a lot at UCL. So thank you very much for your attention, and I'm very happy to answer questions.
So Aroon, many thanks for your brilliant presentation. So I was learning a lot. I forgot at the beginning, as I always do, to announce that you should post your questions into the chat. And while waiting for the questions posted there, I would like to start maybe also with some provocative questions. So when I listened to your presentation, and also when I learned from the tremendous results there, I got the impression that this kind of approach works very nicely for diseases where you have like an inflammatory component, and you can nicely quantify certain biomarkers or lead proteins in the plasma, for instance. But it might not work as nicely with other diseases where it is much more difficult to really establish this genotype-phenotype link. That would be my first question. So how would you approach that? How do you see that actually?
Yeah, that's a critical question. So actually, a critical step in using this approach is to validate that the genetic variants that you study actually have some effect, because not all do. And as you say, in order to do that, you need some kind of biomarker that you can use as a weighting variable to show that the genetic variant has an effect. Now, the ideal weighting variable, if you're taking a drug development approach, is to use variants in the gene that encode the drug target and actually to measure the level of the drug target or the function of the drug target itself. Now, the options to do that have expanded through the development of proteomics, but the limitation is that those are proteins that you find in the circulation. And not all the proteins that you're interested in targeting therapeutically circulate. So one thing to say, nevertheless, there are many drug targets that do circulate, and so there are several thousand to work on in that setting. But ideally, we'd like to know about genetic variants that influence expression or function of proteins in tissues. Currently, we can only do that indirectly through mRNA expression. So GWAS of mRNA expression is really quite advanced through resources such as GTEx, EQTLGen, and so on. So we frequently use mRNA as a weighting variable. But we have to make some assumptions there. Which tissue are we working on? And is that the correct one? And is there a direct relationship between the mRNA expression and the protein, which we're really trying to proxy? And there may not be always. And there are certain conditions and diseases which I think pose difficulties, particularly, for example, neurological diseases,
Yep.
where the targets are within the brain. There may not be a useful or readily accessible proxy. So these are all limitations of the approach, as you very rightly point out. So we won't be able to solve every problem this way, no doubt.
Yeah, but with including maybe also imaging data more and more into this kind of analysis, that might also give some better glimpse into these diseases as well.
Absolutely, yeah.
And then I have also the question on, oh, I actually forgot mine. Maybe there is a question of how well can cis-MR estimate the potential drug effect size?
Yeah, that's one that many people think about. Actually, it's a very good question. So I'll give you an example. We know HMG-CoA reductase excellent target for LDL cholesterol lowering and protection from coronary heart disease. Yet in GWAS of coronary heart disease, probably the HMG-CoA reductase is variants in that gene are probably, I don't know, maybe the 50th or 60th in terms of effect size. So tiny effect. They weren't the first coronary heart disease locus to be found earlier in LDL cholesterol GWAS. But again, not the strongest effect on LDL cholesterol. So I think what that's been telling us is that genetic effects can be very small, but nevertheless, when you make a drug against the encoded target, because the drug's so much more potent in terms of its effect on target perturbation, you can see quite a big effect, useful therapeutic effect on disease risk, even though the genetic effect might be small. So the way that we've been thinking about this is that even finding small genetic effects is important because you can tend to design a drug that can be fairly potent on the target and have a very substantial effect, important therapeutic effect. So I think it's quite hard to say anything more about a genetic variant proxying the size of a therapeutic effect other than don't be put off by a small genetic effect. As long as you can ascertain it reliably, as long as you can be confident it's real, things are worth pursuing at a therapeutic level.
So thanks a lot. I would also have two more questions. I mean, one goes into the direction of my first one. What about diseases where the genetic component is not playing such a tremendous role, like we had in the COVID-19 pandemic, where obviously with the wild type, it was rather age driving the disease, the severe disease. So, but you never know before you actually discover that. how do we avoid running into a maybe false direction, looking for something which you cannot find?
Yeah, so I think so. Two other areas of limitation of this technique that are important to point out is that this is based on using genetic variants in the germline to proxy the effect of drugs. So genetic variation that arises in cancer cells, we can't model using this technique because those are somatic variants. Moreover, we can't model therapeutic targets in an infectious organism because obviously the protein target is typically within the organism. But what we can do using this technique is to model host response. And one of the examples actually is that the genetic variants in the interleukin-6 receptor has been shown to confer risk of severity of adverse COVID-19 outcome. And actually, the interleukin-6 receptor is the target of tocilizumab, and tocilizumab has been proven to be effective in randomized trials in protecting from death in patients with severe COVID-19. So we can actually say something with this technique about host response to infection and the likelihood of a therapeutic target that modulates an overexuberant, for example, if inflammatory response in the host that might have adverse downstream effects from infection. There is evidence that this technique can be used in that context, but not to identify therapeutic targets in the organism as opposed to the host.
Okay, then there's another question in the chat. GWAS point to mechanism prior to disease development. How likely are those the same as we need to target to cure the disease?
Yeah, this is interesting. Quite right. Most GWASs, almost all, identify genetic variants of disease risk, onset of disease, not prognosis after the disease. So there are very few of those. And in fact, one of my colleagues at UCL, Riaz Patel, is undertaking a GWAS of coronary heart disease event recurrence. So the start point is people with coronary heart disease, and you do a GWAS for adverse outcome among those people. One of the difficulties that arises there is something called index event bias, which is whereas in GWASs in general populations or even case control settings, you're not selecting in advance on the presence of disease. You are in those studies that start where the start point is people with the disease, and people with the disease are a sort of biased set of the population, and so you could introduce some degree of bias that's not present in the other types of GWAS or Mendelian randomization analyses, and there are now methods that are now trying to address that. But one thing I think which is important to work out, I don't know why this is that if we've done GWASs of coronary heart disease onset and we come up with HMG-CoA reductase, PCSK9, et cetera, and those drugs are effective in people who've already got the disease. So somehow we are getting, at least in many cases, reliable insights. And we could say the same of rheumatoid arthritis, for example. We're using tocilizumab to treat patients with rheumatoid arthritis, not to prevent its development. Yet the GWASs were done of rheumatoid arthritis risk and onset. So there is useful information emerging from GWASs for reasons that are not entirely clear, that has relevance for people with established disease already. So I don't know why, but I know it's working.
Yeah,
at least in many cases, but maybe not all.
So and I mean those kind of investigations are really great, but they also imply a big change in our in the way we do science and research, because it requires large sums of money, large patient cohorts, and personally I think one would need to really change also the way we did really structure and design research, and also work more together, although it's even becoming more difficult with the UK now, within Europe, yeah. So as a whole, because you need large patient cohorts, you need maybe large consortium, but also a different philosophy in is it still valid to just judge first and last authorship, because these are really big networks you need here. So what is your opinion on your the strategy in the UK? Is that leading to a change in the system?
So I think there is very much an emphasis on large-scale collaborative studies and data sharing. One of the advantages that this particular approach has is that you can work on summary level data, not on individual level data, and share the summary level estimates across jurisdictions without running into difficulties in terms of data sharing across borders or across jurisdictions. So that's one thing. But in terms of other things, this change towards a kind of team science approach rather than a single lab working on a single question, that really is, you're quite right, becoming a paradigm shift in the way biomedical science is operating, and ensuring that there is appropriate credit for people. What's often forgotten, for example, that when you use data from longitudinal cohort studies that have assembled phenotypic data over many, many years, but a genetic study is published and the first and last authors are the people who've done the genetic analysis. All the phenotype collection over the many years, the curation of the cohort, the follow-up, etc., tends to receive less emphasis than it should. So that type of recognition is also important, but also the importance of recognizing people who enable the work. So a lot of data scientists, software engineers, computational scientists, many of whom don't necessarily want to build their own research group and become a PI, but who are absolutely critical and technically superbly expert, very innovative, etc., often don't receive the credit or have the career pathways currently that they should. People have an awareness of this now, but I think more needs to be done to address that.
Absolutely, I completely agree. So that's why I wanted to raise that issue, because it is important for the careers of the young scientists who are joining such an collaborative network, because it will determine their later success also. So I don't see any more questions, but I'd like to thank you again, and I really hope to see you in person at some point, because I would like to discuss that in more detail, maybe.
It would be a pleasure.
Yeah. So thank
we have
you.
one more question, I think. It just came in.
Okay. So the person, I don't know who, was also thanking you for this fascinating talk, and then asked the question: in which direction will the drug development go from multigenetic diseases for multigenetic diseases, and even broader? Could you suggest how we could, with use of more and more data, surpass adverse effects of drugs which have more targets?
So on the first question, which direction is drug development going? I mean, from what we see, the way the pharmaceutical industry is operating now, they are placing very high priority on human genomic data to inform their drug development program. So I think this is going to grow in the commercial sector, is already growing. Most common diseases have multiple genetic loci, but we know we get good therapeutic benefit even when we target one or two loci. We know this from coronary disease. We target HMG co-reductase, PCSK9, etc. We see really substantial effects on coronary heart disease risk. So even identifying a small number of targets from the many that might be out there can be useful. I forgot the last component of the question, which was.
Could you suggest how we could, with use of more and more data, surpass adverse effects?
So we can use this approach to model adverse effects, but only on target adverse effects, not off target actions of compounds or drugs that might act on other targets because they're not entirely specific to the target that we're proxying. So an example of these on target effects was, we showed genetically that taking a statin increases your body weight a little bit and your risk of type 2 diabetes. That doesn't matter because the effect on coronary heart disease is so great, the effect on diabetes is very small. We showed that the first CETP inhibitor drug that was evaluated, torcetrapib, which was found to raise blood pressure, we showed genetically that blood pressure effect is off target, and the more specific CETP inhibitors don't have that effect. So we can use it to help distinguish on versus off-target effects of new drugs. We can't use it to predict off-target effects. That's beyond scope.
Yep, absolutely. Okay, great. Then I hope you enjoyed giving the presentation as well, and
That's great.
I wish you a fantastic afternoon in the UK, and hope
Thank
to
you.
see you soon. Thanks,
Thanks
Aroon.
very much. Thank you, everyone. Bye-bye.
Bye.